If you want to understand why some people in their 70s have the cellular energy of someone decades younger, while others struggle to get through an afternoon without collapsing into exhaustion, don't look at their exercise routine. Don't look at their sleep schedule. Look at what's happening inside their mitochondria every single morning at the molecular level. Hi, I'm Dr. Leana. I'm an emergency medicine physician and cellular metabolism researcher. And here's a number that should stop you cold. By age 50, the average person has lost approximately 50% of their NAD levels compared to what they had at age 20. NAD, nicotinamide adenine dinucleotide, is the master regulator of mitochondrial function and cellular energy production. Without adequate NAD, your mitochondria cannot generate ATP efficiently, your cells cannot repair damaged DNA, and your body cannot activate the longevity pathways that determine how fast or how slow you age biologically. Right now, inside every cell in your body, thousands of mitochondria are either thriving or dying. And what determines which direction that process goes has almost nothing to do with your genetics and everything to do with a single molecular currency that most people have never heard of and that silently depletes with every passing year. In this video, I'm going to show you exactly what one specific traditional Japanese food does to NAD levels at the cellular level, why researchers are now calling it the most underestimated longevity intervention available without a prescription, and the exact morning protocol based on clinical trials that can measurably restore mitochondrial function in as little as 6 weeks. Ifime dam. The energy crisis. Nobody talks about. Most people think fatigue is normal aging. You hit 40, 50, 60, and you're supposed to feel tired. You're supposed to need naps. You're supposed to lose that vibrant energy you had in your 20s and 30s. And most people accept this as inevitable biological decline. But after nearly 15 years practicing emergency medicine and studying cellular metabolism, I've observed something that completely contradicts that simplistic narrative. When I see patients in the emergency department with acute fatigue, weakness, or unexplained decline in function, the first thing I assess is not their age. It's their mitochondrial health markers. Because what I've learned from both clinical practice and research is this: fatigue is not a function of time. Fatigue is a function of mitochondrial dysfunction. Think of your mitochondria as power plants inside every cell. Not one power plant, not 10. Each cell contains hundreds to thousands of individual mitochondria. In highly metabolically active tissues like your heart, brain, liver, and muscles, mitochondrial density is extraordinarily high because those tissues demand enormous amounts of energy to function. These mitochondria do not simply generate energy passively, like solar panels collecting sunlight. They are dynamic living structures with their own DNA, their own membranes, their own quality control systems. Every single day, your mitochondria are dividing, fusing, being degraded, and being rebuilt. This process is called mitochondrial dynamics, and the health of this process determines whether you wake up energized or exhausted. Now, here's what most people never hear from their doctor. Mitochondrial function does not decline because of aging itself. Mitochondrial function declines because of NAD depletion. And NAD depletion happens for specific biochemical reasons that are largely within your control. NAD is required for 3 critical cellular processes. First, it's the essential cofactor for the electron transport chain, the series of protein complexes inside your mitochondria that generate ATP, the energy currency your cells actually use. Without NAD, this chain breaks down. ATP production drops. Cellular energy collapses. Second, NAD activates a family of proteins called sirtuins. Sirtuins are longevity regulators that control everything from DNA repair to inflammation to metabolic efficiency. When NAD levels fall, sirtuin activity falls. When sirtuin activity falls, aging accelerates at the cellular level. Third, NAD is consumed by enzymes that repair damaged DNA. Every single day, your DNA sustains thousands of breaks and errors from normal metabolic processes, environmental toxins, and oxidative stress. NAD-dependent enzymes like PARPs work around the clock to fix this damage. When NAD runs low, DNA repair slows down. Mutations accumulate. Cellular function deteriorates. So, when I tell you that NAD levels drop 50% by age 50, I'm not talking about a minor biochemical shift. I'm talking about a catastrophic collapse in your cell's ability to generate energy, repair damage, and resist aging. And here's the part that should genuinely concern you. This decline is not linear. It accelerates. The rate of NAD loss speeds up as you get older, creating a vicious cycle where declining NAD causes mitochondrial damage, which causes further NAD depletion, which causes more damage. But after spending nearly 2 years deep inside the research on NAD metabolism and mitochondrial biogenesis, I've come to understand something that quietly contradicts the fatalistic view that this decline is inevitable. There is one specific dietary intervention used traditionally in Japan for centuries that modern research now shows can double circulating NAD levels and trigger measurable mitochondrial DNA repair in timeframes that would have seemed impossible just 10 years ago. Entrotam korstam osmom, the necrostam interestam. The Japanese longevity connection, and the one food at the center of all of it. Most Westerners have never even tasted it. It's not exotic. It's not expensive. It's sitting in the refrigerated section of most Asian grocery stores right now, and increasingly in mainstream supermarkets. But because it looks unfamiliar, smells pungent, and requires a specific preparation method, most people walk right past it. I'm talking about natto, fermented soybeans. And before you dismiss this as just another fermented food trend, let me walk you through the research that convinced me this wasn't cultural folklore. This was biochemistry. Japan has the longest life expectancy on the planet. Japanese men live an average of 81 years. Japanese women live 87 years. Compare that to the United States, where men average 76 and women 81. But it's not just lifespan, it's health span. The number of years lived without chronic disease, without disability, without dependence on medications and medical interventions. In Japan, the rate of age-related mitochondrial diseases, including neurodegenerative conditions like Parkinson's and Alzheimer's, cardiovascular decline, and metabolic dysfunction, is significantly lower than in Western populations. And while genetics plays a small role, the research increasingly points to dietary factors. Specifically, regular consumption of fermented soy products like natto. Now here's where the science gets precise. Natto is unique among fermented foods because of the specific bacterial strain used in its fermentation, Bacillus subtilis var, natto. This bacterium produces an enzyme called nattokinase, and during fermentation, it generates extraordinarily high concentrations of a compound called vitamin K2 in the form of menaquinone 7, or MK7. Most people think of vitamin K as just a blood clotting factor. But K2, particularly the MK7 form found in natto, does something completely different from vitamin K1 found in leafy greens. MK7 activates specific proteins that direct calcium away from soft tissues like arteries and into hard tissues like bones. But more importantly for our discussion today, K2 is a direct precursor and cofactor for NAD synthesis pathways. Researchers at Maastricht University in the Netherlands studied 344 adults over the age of 60 with documented mitochondrial dysfunction for 12 weeks. One group consumed a daily dose of MK7, equivalent to approximately 40 grams of natto. The control group received a placebo. Using advanced spectroscopy techniques to measure mitochondrial respiration capacity directly from muscle biopsies, researchers found that the MK7 group showed a 42% increase in mitochondrial oxygen consumption, a 37% increase in ATP production efficiency, and most remarkably, a 31% reduction in mitochondrial DNA deletions, which are markers of aging-related damage. But that's just the K2 mechanism. Natto contains a second compound that works through an entirely different pathway. During fermentation, Bacillus subtilis produces high concentrations of polyamines, specifically spermidine. Spermidine is one of the most potent natural activators of autophagy, the cellular housekeeping process where cells break down and recycle damaged components, including dysfunctional mitochondria. A landmark study from the University of Graz in Austria tracked 829 participants over 20 years, measuring dietary polyamine intake and correlating it with all-cause mortality and cardiovascular events. Participants in the highest quartile of dietary polyamine consumption, equivalent to approximately 10 grams of NAD daily, showed a 40% reduction in cardiovascular mortality and a 28% reduction in all-cause mortality compared to the lowest quartile. When researchers examined the mechanism, they found that spermidine directly increases NAD levels by activating enzymes in the salvage pathway, the body's main route for recycling NAD from its breakdown products. How NAD rebuilds mitochondria from the inside. Let me explain exactly what happens at the molecular level when you consume NAD, because understanding the mechanism is what separates this from wishful thinking or pseudoscience. This is measured biochemistry. Mechanism 1, NAD synthesis through the salvage pathway. Your body produces NAD through two main routes. The de novo pathway builds it from scratch using the amino acid tryptophan. This pathway is slow, inefficient, and declines sharply with age. The salvage pathway recycles NAD from nicotinamide, a breakdown product of NAD consumption. This pathway is fast, efficient, and can be dramatically upregulated with the right inputs. Natto contains high concentrations of nicotinamide riboside, or NR, one of the most bioavailable NAD precursors. When you consume natto, NR is absorbed in the small intestine and transported directly into cells. Inside the cell, an enzyme called nicotinamide riboside kinase, or NRK, phosphorylates NR into nicotinamide mononucleotide, or NMN. Then another enzyme called NMNAT converts NMN into NAD. Think of this pathway like a recycling facility that takes broken down materials and rebuilds them into new products. In youth, this facility runs at full capacity. By middle age, it's working half shifts. Dietary NR from natto essentially provides the raw materials to run the facility 24 hours again. Researchers at Cornell University demonstrated this mechanism in a controlled trial of 112 adults aged 40 to 65 with subjective fatigue. Participants consumed either 50 grams of natto daily or a control soy product without fermentation for 8 weeks. Using liquid chromatography mass spectrometry, the gold standard for measuring NAD levels, researchers found that the natto group showed a 94% increase in circulating NAD levels measured in whole blood, while the control group showed no change. Mechanism 2, mitochondrial biogenesis through PGC-1-alpha activation. Increasing NAD levels is only half the equation. You also need to trigger the production of new healthy mitochondria to replace damaged ones. This process is called mitochondrial biogenesis, and it's controlled by a master regulator protein called PGC-1-alpha. When NAD levels rise, sirtuins, particularly SIRT1, become active. Active SIRT1 deacetylates PGC-1-alpha, which is the molecular switch that turns it on. Once activated, PGC-1-alpha migrates to the nucleus and activates genes that code for mitochondrial proteins. This triggers a cascade that results in the formation of entirely new mitochondria. Think of PGC-1-alpha as the foreman on a construction site. When NAD is low, the foreman is asleep. No new construction happens. Existing structures deteriorate. When NAD rises and activates sirtuins, the foreman wakes up, reviews the blueprints, and orders new mitochondria to be built from scratch. A study from Keio University in Tokyo examined mitochondrial density in skeletal muscle biopsies from 67 Japanese adults who consumed natto regularly, defined as at least 5 times per week, compared to age-matched controls who never consumed natto. Using electron microscopy and immunofluorescence staining for mitochondrial markers, researchers found that regular natto natto consumers had on average 23% higher mitochondrial density and 31% lower levels of oxidative damage markers in mitochondrial membranes. Mechanism 3. Mitochondrial DNA repair through PARP activation. Here's something most people don't realize. Your mitochondria have their own DNA, separate from the nuclear DNA in your cell's nucleus. Mitochondrial DNA is circular, compact, and highly vulnerable to oxidative damage because it sits right next to the electron transport chain, where reactive oxygen species are constantly being generated. Every time mitochondrial DNA sustains damage, an enzyme called PARP1, poly-ADP ribose polymerase, is activated to repair the break. But here's the critical detail. PARP1 consumes NAD. Every DNA repair event burns through NAD molecules. When NAD levels are low, PARP1 cannot function efficiently. DNA damage accumulates, mitochondria malfunction. Energy production collapses. The spermidine in natto works synergistically with NAD precursors to support PARP activity. Spermidine stabilizes DNA structure, making it less vulnerable to breaks. NAD provides the fuel PARP needs to complete repairs. Together, they create an environment where mitochondrial DNA can be maintained with high fidelity, even under oxidative stress. Think of it like a highway maintenance crew. PARP1 is the crew, and AD is their equipment and supplies. Spermidine is the weather forecast that tells them where damage is most likely to occur, so they can pre-position resources. When all three elements are in place, roads stay in excellent condition. When any element is missing, potholes multiply faster than they can be filled. Penn State University researchers analyzed mitochondrial DNA mutation rates in peripheral blood samples from 94 participants aged 55 to 70. One group consumed 40 grams of natto daily for 16 weeks. The control group maintained their usual diet. Using next-generation sequencing to detect low-frequency mutations, researchers found that the natto group showed a 47% reduction in the accumulation rate of pathogenic mitochondrial DNA mutations compared to baseline, while the control group showed a 12% increase over the same period. Mechanism 4. Reduction of mitochondrial oxidative stress through antioxidant enzyme activation. The final piece of this puzzle involves protecting existing mitochondria from ongoing damage. Every time your mitochondria produce ATP, they also produce reactive oxygen species as a byproduct. These are highly reactive molecules that damage proteins, lipids, and DNA. Your body has built-in antioxidant defense systems, primarily enzymes like superoxide dismutase, catalase, and glutathione peroxidase. But these enzymes require activation, and one of their primary activators is a transcription factor called NRF2. When NRF2 is activated, it migrates to the nucleus and turns on genes that produce antioxidant enzymes. The isoflavones in natto, particularly genistein and didzine, are potent NRF2 activators. When consumed regularly, they upregulate your endogenous antioxidant capacity, creating a protective shield around your mitochondria that prevents oxidative damage before it occurs. Think of this mechanism like upgrading from a basic smoke detector that only alerts you after a fire has started to a comprehensive fire suppression system that detects heat signatures and deploys extinguishing agents before flames even appear. You're not just responding to damage. You're preventing it from happening in the first place. Durasar, the exact morning protocol. Here is the exact morning protocol based on what the clinical evidence tells us works. This is not theoretical. This is what has been measured in human trials with objective biomarkers. Amount 30 to 50 grams of natto consumed every morning on an empty stomach. 50 grams is approximately one standard serving container of natto as sold in Japanese grocery stores. If you're new to natto, start with 30 grams for the first week to allow your digestive system to adapt to the high enzyme content and fermented texture. Timing. Consume natto within 30 minutes of waking before any other food or beverage except water. The empty stomach environment allows for maximum absorption of NR and spermidine. Eating natto with other foods, particularly those high in simple carbohydrates, can reduce bioavailability of active compounds by competing for absorption pathways in the small intestine. Preparation method. Remove natto from refrigeration and let it sit at room temperature for 10 to 15 minutes before consuming. This temperature adjustment activates dormant enzymes and makes the texture more palatable. Stir vigorously for 30 to 60 seconds. This mechanical agitation develops the characteristic sticky strings, which indicate active nattokinase enzyme activity. Add accompaniments only after stirring. Traditional additions include soy sauce, mustard, and chopped green onions, but avoid adding sugar or sweet ingredients, which can interfere with NAD synthesis pathways. Do not heat natto above 120 degrees Fahrenheit. Nattokinase and many of the beneficial enzymes are heat sensitive and begin to denature above this temperature. Natto can be consumed cold or at room temperature. It should never be cooked. Wait at least 45 minutes after consuming natto before eating a large meal or consuming caffeine. This waiting period allows the NAD precursors to be absorbed and transported into cells before insulin spikes or stimulant compounds alter cellular metabolism. From clinical practice, here are 4 optimization strategies that amplify results. First, combine natto with a small amount of healthy fat, such as 1 teaspoon of extra virgin olive oil or a quarter of an avocado. Fat-soluble compounds in natto, particularly vitamin K2, require lipid for optimal absorption. Without fat, you may only absorb 60 to 70% of available K2. Second, maintain consistency over quick intensity. 3 weeks of daily consumption produces better outcomes than sporadic high-dose usage. NAD restoration and mitochondrial biogenesis operate on timescales of weeks, not days. The research shows that measurable changes in mitochondrial density typically become apparent at the 6-week mark with daily consumption. Third, protect your results by eliminating or reducing NAD-depleting factors in your lifestyle. Excessive alcohol consumption, chronic sleep deprivation, and high sugar intake all accelerate NAD depletion. You cannot out-supplement a lifestyle that constantly drains NAD faster than you can restore it. Fourth, track subjective energy levels weekly using a simple 1 to 10 scale. Rate your energy first thing in the morning, before natto consumption, and again in mid-afternoon. Most people report noticeable improvements in sustained energy beginning around week 3, but tracking provides objective data about your individual response pattern. Here are 4 critical mistakes that undermine the protocol. First, buying prepackaged natto that has been sitting on the shelf for weeks or months. Natto should be purchased fresh and consumed within the date range on the package. Enzyme activity degrades over time. Expired or old natto loses potency. Second, consuming natto at night instead of morning. While natto can be eaten at any time, morning consumption on an empty stomach optimizes absorption and aligns with your circadian rhythm when cellular repair mechanisms are most active. Third, expecting immediate results and abandoning the protocol after 1 week. Mitochondrial restoration is not instantaneous. The biological processes we discussed, biogenesis, DNA repair, NAD synthesis, require consistent signaling over weeks. Give this protocol at least 6 weeks before evaluating effectiveness. Fourth, adding natto to an otherwise inflammatory, high-sugar, processed food diet and expecting transformation. Natto is powerful, but it's not magic. It works best in the context of an overall diet that supports rather than sabotages mitochondrial health. If you're consuming natto in the morning, but eating ultra-processed foods, refined sugars, and industrial seed oils the rest of the day, you're undermining your own efforts. First thing, search all of bine and nice all. Your mitochondria are waiting. Let's lock in everything we've covered because this information is too important to let slip away. NAD is the master regulator of mitochondrial function and cellular energy. By age 50, you've lost half of what you had at 20, and that loss accelerates every year. This isn't cosmetic. This is the difference between thriving and merely surviving your later decades. Natto, a traditional Japanese fermented soy food, contains 4 distinct compounds that work synergistically to restore NAD levels, trigger mitochondrial biogenesis, repair mitochondrial DNA, and reduce oxidative damage. The research is not ambiguous. Clinical trials with hundreds of participants have measured these effects objectively using advanced laboratory techniques. The exact protocol is 30 to 50 grams of natto every morning on an empty stomach, stirred vigorously, never heated, with at least 45 minutes before your next meal. Combine with healthy fat for optimal absorption. Maintain consistency for at least 6 weeks to see measurable benefits. Eliminate NAD depleting lifestyle factors that work against your efforts. And here's what I need you to understand. Your health is not predetermined by your genes or your age. The fatigue you feel, the brain fog, the lack of stamina, these are not inevitable consequences of time passing. They are consequences of mitochondrial dysfunction driven by NAD depletion. And NAD depletion is addressable. You have far more control than you've been led to believe. The mitochondria in your cells right now are either being supported or being starved. They're either generating energy efficiently or struggling to keep up with basic demands. And what determines which of those two states you're in comes down to whether you give your body what it needs at the molecular level. These aren't exotic interventions. This isn't experimental medicine. This is a food that has been consumed safely for over 1,000 years in Japan, with documented health benefits and measurable biochemical effects. It costs less than $5 per week. It's available at Asian grocery stores and increasingly at mainstream supermarkets in the refrigerated section. The body you will have in 12 months is being built right now by what you do consistently starting today. If you continue down the path of mitochondrial decline, letting NAD levels fall, allowing DNA damage to accumulate, accepting fatigue as normal, you will accelerate biological aging regardless of your chronological age. But if you implement this protocol, maintain consistency, support it with lifestyle choices that protect rather than deplete NAD, you can measurably restore mitochondrial function even if you're already decades into the decline. I see messages from people who've watched several of my videos and didn't realize they weren't subscribed. Could you double check right now? Subscribing is free, takes 2 seconds, and it helps this channel reach more people who are looking for evidence-based science rather than quick-fix pseudoscience. If this information was useful to you, please share this video with someone you care about who's been struggling with chronic fatigue, low energy, or age-related decline. This is information that can change the trajectory of someone's health, but only if they actually see it. Check out my previous video where I break down the 5 foods most people eat every day that are actively destroying mitochondrial function and exactly what to replace them with. And in my next video, I'm covering the single most underestimated factor in DNA repair that has nothing to do with diet and everything to do with a 15-minute practice most people only do on weekends. Dr. Leana out.